I will highlight some of the limitations of a pathway-based enrichment analysis approach. Argument 1 is that biochemical databases are incomplete for metabolomics. This database analysis was done for a metabolomics dataset. Is an example on known obese diabetes mice. The data were downloaded from metabolomics workbench with this study ID. It has 385 primary metabolites, oxalipids, and complex lipids. So on the left side we saw a volcano plot where we also highlight if we found any pathways for these altered compound or not. So blue dots are decreased compound, red dots are increased compound, and yellow are no change on a p-value cutoff of 0.05. And you can see that many empty dots, which represent that there were no pathways, are very significantly affected, and the majority of them are lipids. So if we do it again. I will highlight some of the limitations of a pathway-based enrichment analysis approach. Argument 1 is biochemical databases are incomplete for metabolomics. This database analysis was done for an example metabolomics dataset on known obese diabetic mice. The study was downloaded from metabolomics workbench database, and with this XSN ID, it has 385 primary metabolite, oxalipids, and complex lipids. On the left side we show a volcano plot of significantly altered compound found in the study. Blue dots are decreased, red dots are increased, and yellow means no change. And a filled dot means pathway was found in NCBI biosystems, and empty dots mean pathway, there was no pathway for a compound. And as you can see that significantly affected compound here, many are not having any pathway annotations. Specifically, several lipid species are significantly altered, but since they don't have any pathway notation, a traditional pathway-based approach will ignore these lipids, and they will not be included in final interpretation. On the right side we show how these compounds, known compounds, are represented in different databases. So KEGG, which is a very well-known pathway database, it has the poorest representation, which only 30% of these compounds are present in KEGG. Then if you go to NCBI biosystems, the coverage increases slightly to 173 compounds, and if you go to mesh ontology, it increases to 187. It's about 45%. But as you can see that these biochemical databases, they do not cover all of the compound, known compound, that we have in our high quality metabolomics dataset, and these not covered compound will be ignored in a pathway based approach and further interpretation. So in all the efforts we spend on identification of compound with better computational algorithms and so on, and finally if we go to a pathway based enrichment analysis, those compounds that have been identified using efforts will be ignored because these compounds are not present in any of these biochemical databases. Argument second, pathway definitions are manual and vary across different databases. The graph shows here pathway counts in 5 major pathway databases: KEGG, HMDB, WikiPathway, Reactome, and Metasync. There is a 5-fold difference between pathway count of KEGG and Metasync. This raises a question that maybe they're using different pathway definitions, and there is no consensus among pathway definitions in these databases. Even for a well-known pathway map such as TCA cycle, the maps are different. For example, Metasync do not have pyruvic acid. In here, Metasync do not have pyruvic acid, whereas KEGG has it. As some small molecule pathway database has it. But overall, you see that KEGG has way more number of compound than Metasync, whereas Reactome also have additional compounds for TCA cycle. And this suggests that pathway definitions, even for well-known pathway, are varying from one database to other, and therefore p-values calculated using these definitions will be different because of use of a different pathway database. Argument 3. Pathway definitions are overlapping. This pie chart shows frequency of pathway maps annotations for different compounds in the NCBI Biosystems database. We can see that almost half of them have only one pathway annotation, but more than 30% compounds, little more than 30% compounds have 4 or more pathways. So there is a huge overlap among pathway definitions in these biochemical databases, and this creates two major problems. Number one, it increases the family-wise error rate for hypergeometric test, that we are using the same compounds, however repeating them for various pathway definition. Second problem is interpretation, that for the same compound there could be 10 pathway maps generated, and the interpretation become harder than which pathway was actually impacted. Argument 4. Hypergeometric or Fisher exact test is inappropriate for metabolomics. This test was often used in genomic research. It's okay to use this test for genomics because we know the total number of genes in a genome, so the size of background database is a fix. And for any given genomic assay, whether it's a transcriptomics assay, RNA-seq, or protein, all the genes will be covered in a background database. However, in the metabolomics, we don't know the size of a background database, and is not fixed. And if you use existing biochemical databases as a background database for hypergeometric test, our analytical assays covered several compounds that are not part of these databases yet. So here we have detected metabolite D, and the D covers several other compounds that are not yet covered in N. And size of a background database has a huge impact on the p-value we can get for pathway maps. So here, if you use a larger database. So x-axis shows database size, and as you go from a smaller to larger database for the same pathway, and some same pathway of 10 compound, which 3 were found to be altered in experiment, we will get very small p-values just by changing the size of database, a background database. We have also seen growth in pathway databases that because of the continuous manual curation process. Here shown pathway maps included in MetaSeq database over last 15 or 16 years. As these definitions are included, more compounds are included in the background database, and with every update, if you rerun the pathway enrichment using a biochemical database, you will get different p-values for the same pathway. So a statistical method such as hypergeometric test, which depends on a background database and is not appropriate for metabolomics because n is unknown for metabolomics. Argument 5. Background database size is not defined for metabolomics. What should be a background database for calculating a hypergeometric test? But there are several options, and it's shown in the previous slide that if you use a smaller data set, you will get larger p-values, and if you use a larger database, you will get smaller p-values. So which database we should use, like entire HMDB, which almost 110,000 compounds, or compound with pathway annotations, or compound with reaction annotations, around 4,000 of them for human, or compound with literature that whatever we detect for human blood, we can use all of that, which is about 15,000, or we can say we detected known compound, which is between 500 to 1,000, or detected all compound, which is around 3,000, or 3,000 to 8,000 for human blood specimen. So there is a huge variation in these different background databases, and which one should be used for metabolomics is not defined. So there are major problems in pathway-based enrichment analysis. As summarized in this slide, that not all metabolites from a pathway map are present in a metabolomics dataset. Not all detected metabolites have pathway annotations. Pathway boundaries are arbitrary and overlapping. Pathway maps vary across biochemical databases, and background database size is varying over time for a hypergeometric test. What's the alternative could be is we propose as ChemRICH, a chemical similarity enrichment analysis, which is a pathway independent method that uses all identified compound and uses non-overlapping set definitions that does not depend on any background database for calculating p-values for each set. So what are alternative set definition and statistics that we can use for metabolomics enrichment analysis? So what are alternative set definitions? Alternative A is metamap clusters. All the known metabolites, the idea is that all the known metabolites can be mapped into a metabolic network graph, and using a cluster detection algorithm we can define clusters within that network graph and use those clusters as set definitions. Metamap uses known biochemical reactions among metabolites, and also uses chemical similarity mapping to map metabolites that do not have known any reactions. Then it creates a network graph that can be visualized in Cytoscape. Visualization of such a graph highlights different metabolic clusters or modules, such as organic and TCA cycle compounds, free fatty acids, and nucleotides, and etc. These clusters can be detected automatically using known cluster detection algorithm. They are readily available within Cytoscape using a plugin known as Cluster Maker. It's straightforward to detect these clusters. However, there are two limitations. One is that we still need to manually define cluster label. So this cluster maker algorithm or tool will detect this cluster, but we still need to zoom in and put a label on it, such as amino acid or amines. Then also the network graph is created using a similarity threshold of 70%, and the threshold has impact on the size or its definition of clusters. That on a higher threshold we might get more clusters, and on a lower we get a low number of clusters. Alternative B could be chemical similarity clusters. The idea is to map all the known compounds into a chemical similarity tree, then use a tree cutting algorithm to define clusters. So here in this tree model, blue dots are decreased compound, red dots are increased compound, and darker lines are cluster boundaries that were showing boundaries of enriched cluster, and gray are other compounds. And you can use a distance metric such as Tanimoto coefficient among metabolite to calculate this tree. It is straightforward to create these tree models and also to define these clusters using a tree cutting algorithm such as dynamic tree cut package in R. The limitation is the cluster label, that although we have these nice clusters, we still need to zoom in and define that these are lipids and these are some other chemical classes. Alternative C, chemical ontologies. I want to highlight two major ontology database for metabolites. Number one is MESH, medical subject heading ontology, that has almost 110,000 compounds in it. So here is an example for carbohydrate class, that carbohydrates are divided into monosaccharides, and monosaccharides are further divided into hexoses and ketoses and so on. These terms were extracted from biomedical literature from PubMed, and they have been linked with PubMed. So these mass ontology allow automated text mining if needed. The second ontology is lipid maps ontology, that has almost 50,000 compounds. It's in one of the most comprehensive classification for lipids. However, there is a limitation here, that not every metabolite we detect is covered in MESH or lipid map ontology. But these ontology provides labels for compound classes that can be used for chemical similarity clusters. What could be alternative to hypergeometric test? We suggest that KS test is a better statistical method for metabolomics enrichment for two reasons. That KS test does not depend on a background database, and it doesn't need any p-value cutoff. It can use all the compounds in a metabolomics dataset to calculate set level enrichment p-values. So the table shows a comparison between different tests for enrichment. That Fisher and Hypergeometric test requires a background database and a p-value cutoff. Binomial is independent of a background database, but it will require a p-value cutoff, whereas KS test does not depend on a background database and it doesn't need a p-value cutoff. So KS stands for Kolmogorov-Smirnov test, is a non-parametric test of the equality of continuous one-dimensional probability distribution that can be used to compare a sample with the reference probability distribution. The KS test was used in the seminal paper for gene set enrichment analysis that has been cited by over 11,000 times so far. And basic idea is to compare the distribution of p-value to find the significance on the set level instead of count of affected compound on a p-value cutoff. ChemRICH combines medical subject heading, mesh, chemical similarity, and KS test to build a new enrichment analysis approach for metabolomics. ChemRICH has two components, a pre-compiled ChemRICH database that has almost 91,500 unique structures linked with 2,768 mesh classes. Each compound is associated with a PubChem CID, Smiles code, mesh ID, and a fingerprint. Fingerprints were calculated using RCDK package. For a metabolomics dataset, first we do a lookup search for metabolite name, CID, and Smiles code. If they are already in the ChemRICH database, we will retrieve their mesh annotations. If they are not in ChemRICH, we will calculate an approximate mesh ID based on chemical similarity to the closest similar compound. If compounds are not in the ChemRICH database and they are not similar to any other compound having a mesh annotation, we will look if the leftover compound are making a new cluster. The new cluster, the mesh classes, chemical clusters, and new classes are then feed into a non-overlapping class estimation function that gives one compound one class annotation, and that is used as an input for KS test, where it takes p-value and effect size provided by a user into account and calculates class level p-values, and those are visualized into a ChemRICH impact plot, where we also use the Tanimoto similarity among compounds in a hierarchical tree to calculate an x-axis order for chemicals. This flowchart shows precise steps in ChemRICH analysis for a metabolomic study. That we will start with parsing the Smiles code, and during the parsing we will estimate some class labels just by the structure of Smiles, and then we will do the ChemRICH lookup. So starting is 385 compound, Then we will do the lookup, and we found 151 already in mass database. Then for remaining 166, we did the Tanimoto analysis. We could map almost 147 out of that. Then 19 were not found to in any clusters. So we detect if there is any new cluster among them. No. So none of them were part of any new cluster. Then we lower the threshold to 0.75 in case 90 was too stringent, and maybe just by lowering we can map some of the compounds to approximate mass class. So we could map 14 compounds to mass notations. Remaining pipe compound were two distinct, that they were not similar to any other compound, and they need to be reported individually. All those class estimation from SMILE, regular expression search, chemical ChemRICH lookup, Tanimoto similarity, mass estimation, new cluster, and lowering the Tanimoto similarity threshold to 70% is feed into a known overlapping class annotation estimation. And after that, to calculate the KS test, sets having more than 2 compounds, so at least 3 compounds will be included in KS test. And all the metabolites are feed into a similarity matrix, and using to calculate a hierarchical clustering tree, and the order of that tree is used for calculating x-axis in the enrichment impact plot. And with that, the algorithm ends, and impact plot plus result of the KS test are exported back to your user. Using the ontology chemistry clusters to compute p-values for significant metabolite differences. So for the example study on non-obese diabetic mice, when we did the ChemRICH analysis, this is the result shown as a impact plot, where we have negative log of the p-value on the y-axis and cluster order on the Tanimoto similarity tree. And we can see that all the known polar compounds are clustered on the right side of the plot, and all the polar compounds are clustered on the left side of the plot. We have updated the script that instead of using the cluster tree order, it now visualizes x-axis based on median x log p, which is lipophilicity of chemicals, and lipophilicity correlated with cluster tree order. But use of lipophilicity has advantage that we will always have polar metabolites on the left side of a plot and known polar on the right side of the plot. And here red means most of the compounds are increased in this cluster. Blue means most of the compound were decreased in this cluster. Size of the cluster means how many compounds we have in this class. If a cluster is purple, means it's a mixed pattern within the cluster. And we saw that ChemRICH highlighted chemical classes that were originally missed by the authors of the study, such as sugar acid were missed by the authors, and we have also several other classes that could have been discussed for that paper. So ChemRICH has advantage that it highlights chemical classes that are relevant for a metabolomic study, and these can be interpreted further. tabular output includes cluster names, cluster size, p-value, adjusted p-value by FDR, how many total compounds are changed, how many are increased and decreased within that cluster. ChemRICH is available online at www.chemrich.us. You will be redirected to other URL where the ChemRICH app is hosted. I wrote the ChemRICH app in OpenCPU framework for R. The input file has this structure that the first column should be compound names, second column should be INCHI keys, third column should be PubChem IDs, fourth column should be SMILES code, and then we have p-value and fold changes. At the moment, ChemRICH allows only one statistical comparison. I'm writing a R function that will allow, if you have more than one statistical comparison, you can perform ChemRICH analysis in one step. Output are a tree structure, an interactive cluster plot where you can see what chemicals are inside this cluster. Then we have an interactive compound table and a cluster level table. Results can be downloaded as PowerPoint, XLS, PNG, and PDF formats. We also added, in case user want to use their own class information, ChemRICH is doing approximation of mass classes, and several times you may not agree with that. In that case, you can update the class information and using ChemRICH class tool, which takes input of chemical classes by user, and file format is exactly same, but you have additional column class where you can define your own classes, and then ChemRICH will calculate the p-values for that class and create this impact plot and organize these classes by lipophilicity and p-value. We have made ChemRICH available as a Docker image, and source code is available at my GitHub website. The best way would be to download the Docker image and run it. It's straightforward to do it. Otherwise, it's an OpenCPU app, and you need to install OpenCPU and some other R packages to run ChemRICH locally. In conclusion, there are several advantages for using ChemRICH for metabolomics. It maps up to 95% of the known compounds in a metabolomics data set into chemical classes. The clusters are non-overlapping, so there is no problem of repetitive analysis of same metabolites for multiple sets. It uses a background database independent statistics, so the p-values that are generated today will be reproducible after 6 months, because it uses the data set itself as a database. It doesn't depend on a biochemical database. It can map compounds that are not yet in any database, such as in silico compounds. In silico compounds have SMILES code calculated. You can use those SMILES code to map them into chemical classes. And this way, approaches such as in silico spectra predictions, when they identify any chemical compound in a metabolomics dataset, those compounds can be easily included in enrichment analysis. It utilizes existing knowledge from chemical ontologies to enable straightforward literature mining. All the class label names by mesh are extracted from PubMed. So if you search those names into PubMed database, you already find the relevant publications. It allows identification of new chemical clusters. So if MASH doesn't cover, and because we do untargeted metabolomics, there could be new clusters that are not in MASH yet. And so ChemRICH will be able to identify those clusters, and if they are important for a study, it will highlight them as a new cluster in the impact plot. Cluster impact plot visualizes chemical diversity, lipophilicity, which is helpful in biological interpretation of metabolomics data set. Overall inclusion of well-known chemical classes, and we have as well a room for clustering of other chemical classes. ChemRICH has been published in this Journal Scientific Report. With that, I would like to thank WCMC and NIH for their support to the West Coast Metabolomics Center. Thank you.